Now, talking about the process, the enzyme that fixes carbon dioxide to RUBP in the Calvin cycle is the enzyme Rubisco or the ribulose bisphosphate carboxylase or ribulose bisphosphate oxygenase. An important property of Rubisco is that it has the ability to catalyze both carboxylation or the addition of carbon dioxide into a molecule and oxygenation which adds up oxygen. Hence the name ribulose bisphosphate carboxylase or oxygenase. When oxygenation happens, it results into a process called as photorespiration, wherein it decreases the efficiency of photosynthesis because what it fixes in the C3 cycle or in the Calvin cycle is not carbon dioxide but oxygen. Therefore, there is a loss of carbons in the cycle. Now, when does this happen? When does photorespiration take place? Photorespiration or oxygenation happens when plants are exposed to an environment which has greater amount of atmospheric oxygen than carbon dioxide, wherein differences in the amount of oxygen and carbon dioxide in the atmosphere is also affected by the temperature. When in high temperature, the amount of atmospheric oxygen tends to become more than the atmospheric carbon dioxide. Thus, it increases the chance that Rubisco fixes oxygen instead of carbon dioxide. The biological importance of photorespiration is not yet known. However, there are some probable importance of this, wherein photorespiration is believed to prevent the damage of photosynthetic apparatus of plants that are placed in light intensity and low carbon dioxide concentration because stomata have to close to prevent water loss. Luckily, there are some plants that are capable to adapting to photorespiring conditions. These plants have developed an additional mechanism with the C3 pathway or the Calvin-Benson pathway to efficiently concentrate carbon dioxide. These are the C4 or the Hatch-Slack pathway, and the other one is the CAM pathway or the crassulacean acid metabolism. Now, for C4, this mechanism of adaptation of plants makes use of a morphological adaptation, wherein these plants, there is a presence of another type of cells aside from the mesophyll cells, which are used by the C3 plants, or the plants that make use of the Calvin cycle. These cells are used these cells or the additional cells are called as the bundle sheet cells. So aside from mesophyll cells, they also have these the bundle sheet cells. In the process, the atmospheric carbon dioxide is incorporated into a 3-carbon compound to make the first stable compound of the process, which has 4 carbons, hence the name C4 pathway. In this incorporation of carbon dioxide, there is an involvement of the enzyme called as PEP carboxylase. And the plants that utilize this mechanism are those plants that live in dry and hot environments. And examples of these are the corn or maize and then the sugarcane. So in the process, again, this is the mesophyll cell and this is the bundle sheet cell, wherein the fixation or the fixing of carbon dioxide takes place in the mesophyll cell. The initial step of the cycle is that the carbon dioxide is being fixed into this 3-carbon compound, which is called as the phosphoenol pyruvate via the enzyme called as the phosphoenol pyruvate carboxylase. Meaning to say, it only fixes carbon dioxide instead of oxygen. And with that, there is a formation of a 4-carbon compound which is called as the oxaloacetic acid or oxaloacetate, which is then immediately reduced into the malic acid, which still has 4 carbons. And then this malic acid is going to be transported into the bundle sheet cells via the channel called as the plasmodesmata. So the malic acid is now found in the bundle sheet cell. Now in the bundle sheet cell, this is where the Calvin cycle or the C3 cycle takes place. The malic acid is going to be decarboxylated. When we say decarboxylation, this is the process of the removal of the carbon, specifically in the form of carbon dioxide. So the carbon dioxide fixed in the mesophyll cell is going to be decarboxylated from the malic acid, and this carbon dioxide is going to be given into the normal Calvin cycle, therefore producing now sugars or carbohydrates in the process. And the remaining 3-carbon compound, which is called as the pyruvate, is going to go back into the mesophyll cell and is going to be phosphorylated via ATP to make now the phosphoenol pyruvate, which is the molecule that accepts carbon dioxide in the mesophyll cell. The purpose of concentrating malic acid here is to make sure that what is fixed by the Rubisco in the C3 cycle or the Calvin cycle is only carbon dioxide, not oxygen. And of course, one of the advantages of this one is that it reduces stomatal aperture or the opening of the stomata. Therefore, it prevents the damage of the photosynthetic apparatus. And another advantage of this one, aside from to the C3 plants, is that they can operate at low carbon dioxide concentrated environments, and they can photosynthesize more efficiently at high temperatures. And last one, the Hatchlock pathway or the C4 pathway is suppressing photorespiration or the involvement of oxygen in the process. Aside from C4, we also have the camp plants. Camp plants, on the other hand, does a behavioral adaptation. These plants are those plants that are found in dry environments such as deserts, and examples of these plants are the cacti and pineapples. In the morning, to prevent water loss, the stomata of these plants have to close. At night, since it is colder, stomata open to fix now the atmospheric carbon dioxide. The carbon dioxide is incorporated with phosphoenol pyruvate with the help of the enzyme PEP carboxylase or phosphoenol pyruvate carboxylase. This process produces a 4-carbon compound which is then reduced into malic acid. The malic acid is then stored in the central vacuole of the plant, and then there the malic acid accumulates, so does carbon dioxide. So again, this is where the first step happens since they only open their stomata during the night, so they can only fix carbon dioxide at night time. And again, the initial process involves the incorporation or the fixing of carbon dioxide to phosphoenol pyruvate, forming now a 4-carbon compound which is called as the malic acid. And then this malic acid is going to be stored in the central vacuole of the plant, making now the plant acidic because of the presence of a lot of malic acid. Now, during the day, the stomata close, preventing now the water loss and further take up of carbon dioxide. In the morning, the vacuole deacidifies, so it releases now the malic acid stored in itself. And after it's released, the malic acid is going to be decarboxylated. And again, decarboxylation means the removal of the carbon, specifically in the form of carbon dioxide. So, because the stomata are closed, the release of carbon dioxide cannot escape. So it is only going to be fixed in the Calvin-Benson cycle, therefore producing now sugars or carbohydrates. The remaining 3-carbon compound, which is in the form of pyruvate, is going to be synthesized to form starch. And eventually, this starch becomes a source of PEP or phosphoenol pyruvate, and this phosphoenol pyruvate is ready again to fix another set of carbon dioxides that enter during the nighttime. So of course, advantages of camp plants is that they are having a maximum water use efficiency. It's because they close their stomata at daytime to prevent water loss, and they operate in extreme and dry environments, and of course, it suppresses photorespiration. In summary, photosynthesis in C4 plants are happening, in C4 plants, photosynthesis happens in 2 types of cells, which are the bundle sheath and mesophyll cells. On the other hand, for camp plants, it only happens in mesophyll cells. And the type of separation of the process of photosynthesis is spatial separation because the process is separated based on the structure. Then also for camp plants, it is a temporal separation because the separation of photosynthesis is based on the time of the day and the daytime and then the nighttime. And then in the process, the initial stable compound is a 4-carbon compound, the same with the 2, still a 4-carbon compound, which is in the most case, it's a malic acid. And then the PEP carboxylase or the enzyme that fixes now the carbon dioxide for C4 is active at daytime, while for camp plants, since they fix carbon dioxide at nighttime, so PEP carboxylase is active at nighttime. And again, examples of C4 plants are sugar canes and corns, and for camp plants, we have the cacti and pineapples. Now, the G3P molecules produced in the Calvin cycle are assembled to either sucrose or starch, not glucose. Remember this, that the products of photosynthesis are either sucrose or starch, not glucose. Now, both of these, again, are formed from the G3P molecules produced in the C3 pathway or the Calvin-Benson cycle. The fate of G3P molecules, whether it would be synthesized to make starch in the chloroplast or sucrose in the cytosol in the cytoplasm, is determined based on its concentration and the concentration of orthophosphates at the cytosol. If the cytosol contains high concentration of orthophosphates, the G3P molecules go there to be synthesized as sucrose. On the other hand, if the orthophosphate concentration in the cytosol is low, G3P molecules remain inside the chloroplast to be synthesized into starch. So you have to take note that the products of photosynthesis are actually the starch and sucrose, not glucose. But technically, the products of Calvin cycle or C3 cycle are the G3P molecules. Now, in summary, for the light and the carbon reactions, the light reactions are carried out by the molecules in the thylakoid membrane, and its function is to convert light energy to make ATP molecules and NADPH molecules, and it utilizes photosystems and electron carriers, which are again found on in the membrane of the thylakoid. And light reactions have these two electron flows, which are the cyclic and the non-cyclic electron flow. On the other hand, for carbon reactions, it takes place in the fluid of the chloroplast, which is called as the stroma, and it uses the ATP molecules and NADPH molecules from the light reactions to synthesize glyceraldehyde 3-phosphates, in which these G3P molecules are later on synthesized to make now the final products of photosynthesis, which are the sucrose or starch. And again, for carbon reactions, the carbon reactions utilize enzymes and carbon dioxide acceptors. And lastly, it has these three pathways, which are the C3, which is the most common of all. And then for the adapting mechanisms, we have the C4 and then the CAM pathway.